Hello. This is the first of our Zoom um, lectures. And what I'm going to do is I'm going to share the slides. Let me do that. Turn this off. And we're going to look at mitosis. So this is just a short video looking at mitosis. Okay. So in this video, we're going to talk about cells and we're going to talk about chromosomes. We'll talk about the process of mitosis. A lot of that's going to be review for most of you guys, but we will talk about the role of microtubules in mitosis, which might be a little bit new. Okay. So remember that the cell, of course, is going to be surrounded by a membrane. That's in order to keep things in and prevent things from have selective permeability, so we can control what comes in and out. Cells will also contain organelles. Prokaryotes mostly will contain just basically ribosomes, but eukaryotes will have a number of organelles, including the nucleus, which will have a role to play in cell division. Some cells have cell walls, particularly prokaryotes like eubacteria or archaeobacteria will have cell walls. In the eukaryotes, you have plants and you have fungi that will also have cell walls. Inside of the cells, some bacteria will have an intracellular membrane, which is a membrane that goes through the cell. Generally speaking, what happens is those cells will use that membrane in order to do photosynthesis. Eukaryotes will have intramembrane system that will connect the nucleus to other parts of the cell. So like you have the endoplasmic reticulum, you will also have other things like vesicles that can come off of the endoplasmic reticulum. You have a Golgi apparatus, and those can be basically parts of the cells that can communicate with the membrane and with other parts of the cell. Prokaryotic cells are very simple. They will have ribosomes to translate proteins. They will have, generally speaking, one chromosome that's in the middle of the cell. They may also have plasmids, and plasmids are small circular pieces of DNA that replicate independently of the main chromosome. That one circular chromosome basically is, because it's a circle, it doesn't have a beginning, it doesn't have an end, it doesn't have a centromere or a telomere, it is just one chromosome. Prokaryotic cells include bacteria and archaeobacteria, which archaeobacteria are an ancient form of bacteria that have certain differences that you'll learn about in microbiology. Eukaryotes are everything else, and what defines a eukaryote is the presence of a nucleus. The nucleus has the DNA inside of it in the form of chromatin. That also has the ability to selectively be permeable, so certain things can come in and certain things can go out, but it protects the DNA by keeping out a lot of stuff that goes on in the cell. Eukaryotes also have organelles like mitochondria and chloroplasts, and those organelles have their own DNA and they have their own ribosomes. The nucleus contains the DNA organized into chromatin. That DNA gets transcribed into RNA inside the nucleus. It gets translated into proteins in the cytoplasm. But there is some protein in the nucleus because you need some proteins in order to organize the DNA and to transcribe the DNA. So the genetic material for both prokaryotes and eukaryotes is in the form of DNA, in the form of chromatin, which is basically the non-condensed form of the DNA, or chromosomes, which are which is chromatin, but chromatin, generally speaking, that's been condensed into a more compact form. Okay, bacteria have one chromosome that's often circular, usually, and eukaryotes will have usually double-stranded DNA in the form of several chromosomes that are linear. And because they're linear, they contain a centromere, which is basically the area that's going to connect to the spindle during replication. And they contain a telomere, which is the end of the chromosome. In both cases, for both eukaryotes and prokaryotes, the purpose of the chromosome is to transmit genetic information and to allow that genetic information to be transcribed into RNA and translated into proteins. And the proteins will do the work of the cell. So cell division in terms of mitosis. Mitosis is used in single cell organisms for asexual reproduction. So if you're a single cell organism like a bacteria or an amoeba, which is a protozoa, or a yeast, which is a fungi, you will use mitosis to asexually make a copy of yourself in the form of two daughter cells. And those two daughter cells can go off and live independently and form new yeast cells or amoeba cells or bacteria cells. For a multicellular organism, mitosis is used to basically go from a single cell to a multicellular organism. And as an example, humans start off as a single fertilized egg, and then we end up with several trillion cells by the time that we're adults. Okay? So briefly looking at prokaryotic cell division, we call this binary fission, but it's a form of asexual reproduction. Because they have one circular chromosome, when that circular chromosome replicates in S phase, it makes two chromosomes, and those are usually attached to different parts of the membrane, so that when the cells then go through the process of dividing, those two chromosomes can be placed into two different cells. So what then happens is the cell elongates, the two chromosomes are driven apart, and then you form a contractile ring in the middle of the cell using cytoskeletal proteins. In this case, a cytoskeletal protein called FTSC, though there's other proteins involved as well. That then basically allows the cells to move apart from one another as two daughter cells. You can also build a cell wall in between these two cells if that organism has a cell wall. Okay? We think of prokaryotic cells as one-end cells. In other words, they only have one copy of each gene inside of their cell. We also call that haploid, Haploid, haploid. So haploid and one end are the same things. So when they replicate, they copy each of those genes, but this is still considered to be a one-end cell with a replicated chromosome. Okay. At the end here, each of these daughter cells has a complete set of DNA, but each gene basically is on the chromosome only once. There are some exceptions to that. There's a weird little bacteria called radiodurans. I can't think of its first name now. There's a weird little bacteria that basically is resistant to radiation. And one of the ways it becomes resistant to radiation is by having several copies of each gene, so that when it has a mutation in one copy, it can use the good copy to replace that bad copy. But that's a really big exception to the general rule that prokaryotes are one-end cells. Okay, in eukaryotes, the parental cell and the daughter cells are diploid. In other words, they have two copies of every gene, and we call those two copies alleles. So diploid and 2N mean the same thing. Okay, so our cells are 2N or they're diploid. And when we think about that as being one copy from mom and one copy from dad. Okay, in meiosis, we make one-end cells that only keep one of those two copies. Okay. Each cell produces, during mitosis, each cell produces two identical daughter cells, and we go through these sequential phases of the cell cycle. Interphase, which starts with G1, goes to S phase where the DNA is replicated and ends up in G2 phase where the cells prepare to be divided. G1 phase basically is a growth phase. The cells will just basically at that point be growing and getting signals to tell them whether or not they need to divide or they should divide. S phase is where they replicate the DNA, and G2 phase is where they prepare to go through M phase or the division phase. Prokaryotic cells will do this as well. They also have G1 phase, S phase, and G2 phase. Their stages down here are a little bit different just in terms of what will happen there. The next 4 phases for eukaryotes are prophase, metaphase, anaphase, and telophase. Okay? And we'll talk about each of these in turn, but you remember from previous stuff that you've studied, prophase is where the cells will, the DNA will start to condense into the chromosomes. Metaphase is where those chromosomes line up in the middle of the cell. Anaphase is where those chromosomes will move towards the end of the cells. And telophase is where the one cell divides into 2 cells. Okay? Now, we talked about this is you go through G1 phase, S phase, G2 phase, and then M phase. M phase is really the shortest part of that cycle. Most cells spend the majority of their time in G1 phase, and in fact, as an adult, most of the cells in your body are actually in G0 phase. They're specialized cells, and they no longer go through the cell cycle. They simply stay in G0 phase. They can make proteins, they can metabolize, but they no longer will go through S phase. All of the organs in your body, however, have stem cells that can continue to divide. And those cells that continue to divide will replenish the cells that then will die off at the end of their lifespan. So the stem cells are the ones that continue to go through G1, S phase, G2, and M phase within the human body as an adult. Okay? As we go through the cell cycle into M phase, you form this thing called the spindle. And I'm going to move forward a slide to show you that. So here is the spindle. The spindle basically has 2 centrioles, or microtubule organizing centers on both sides, in the form of this thing that's called the centrosome. So you've got, I should say, 2 sets of centrioles on both sides in this centrosome. And then you have microtubules going off in every direction. On the back side, you have these astromicrotubules. And the astromicrotubules are basically the things that make the spindle positioned in the right place so that it stays where it needs to be throughout the process of M phase. Okay? You also have these ones over here, these golden ones that come off from the other side of the spindle that are called polar microtubules. And these polar microtubules basically will not connect to the chromosomes. They will overlap with one another. You can see the overlap over here, and you can see the overlap there, but they don't connect to the chromatin. What those will do is they will elongate the spindle and elongate the cell to make more room for the chromosomes to separate. You also have what are called kinetochore microtubules, and kinetochore microtubules connect to the kinetochore, which is basically at the centromere, or this area that's basically kind of this constricted area that connects between those 2 sister chromatids. The centromere is not always in the center of the chromosome, but it is the place where you form you put a set of proteins on top of this that's called the kinetochore. The kinetochore set of proteins is what connects to the kinetochore microtubules. And they're connected to this part of the spindle on this side and this part of the spindle on that side. The kinetochore microtubules will shorten as time goes on, and as they shorten, they will bring along the sister chromatids, one going to one centriole and one going towards the other centriole. What drives that elongation and what drives the ability of the chromosomes to move are motor proteins. So you have two sets of motor proteins here as well. The polar microtubules, the ones that overlap but don't touch the chromosomes, they are pushed apart from each other by a motor protein that's called kinesin. And kinesin basically will cause them to move apart. And as they move apart, they elongate the cell so that the chromosomes have more room to separate from one another, and so that you can make two cells out of one cell. Dynesin is another, or dynein, not dynesin. Dynein is another motor protein. And what it will do is it will move the chromatin, move the two sister chromatids towards the centrosome as you get into anaphase, as you go from metaphase to anaphase. At that point, the two sister chromatids will separate from one another, and they'll go towards the two centrosomes, moving towards the ends of the cell. You want that to be done evenly so that you have one sister chromatid on one side and one sister chromatid on the other side. As the dynein molecules move the chromatids away from one another, the microtubules, the kinetochore microtubules, will shorten. They'll rapidly depolymerize after the chromosomes have started to move. So they get shorter as the polar microtubules get longer. Okay, so if we go through the cell cycle, so we start with interphase, which is basically where we see the growth period with G1. The sister chromatids are being formed in S phase. So one chromosome is replicated into two sister chromatids. And in G2 phase, you prepare for division. As you go through this, the centrosome, which contains the two centrioles, is going to be duplicated. Okay? That's going to start to allow you to form the centromere as you start to then go into M phase. As you go into prophase, the spindle is starting to form. So the centrosomes are moving apart. You're starting to see formation of microtubules between them to form the spindle. Okay? The chromosomes are starting to condense. So now you're starting to see this X form, which is really two sister chromatids together. Okay? Two sister chromatids are identical to one another. They had the same genes in the same places. They had the same alleles on both sides of that sister chromatin. They are different than homologous chromosomes. Homologous chromosomes are the chromosome that you get one from mom and one from dad. Okay? When we think about mitosis, we think about sister chromatids. When we think about meiosis, we think about homologous chromosomes. And then later we think about sister chromatids again. As you start to see those chromosomes condensing, at the centromere, you start to see a buildup of proteins to form this thing that's called a kinetochore. And the kinetochore basically are the proteins that will connect the chromosome to the spindle, to the kinetochore microtubules on both sides of that spindle. Okay? The nuclear membrane starts to fragment at that point, and the nucleolus disappears. So if you want to see a difference between interphase and prophase, in prophase you start to see the chromosomes condense, and you see the nucleolus disappear. Okay? The next phase is metaphase. And in metaphase, what happens is the sister chromatids line up on the metaphase plate with the kinetochore microtubule connected on one side to this kinetochore and the other kinetochore connected to the spindle on the other side. Okay? Realize here that your sister chromatids are what's lining up. So you can have, you see the two purple ones here? Those are homologous chromosomes. The two green ones are homologous chromosomes. They line up next to each other with the two sister chromatids facing in opposite directions. Okay? Between metaphase and anaphase, the thing that's going to happen is that the sister chromatids will separate from one another. The sister chromatids are joined together by a protein that's called cohesin. And cohesin basically connects them all along their length, but especially connects them really well at that centromere. So once both sides of the sister chromatids are connected to the spindle, and we'll talk about how this works in class a little bit more, but both sides are connected to the spindle kinetochores, or I'm sorry, the kinetochore microtubules. Then what happens is you have a cleavage of that cohesin by a protein that's called separase. Okay, that's a signal to allow the sister chromatids to separate from one another. There's a lot of tension on those sister chromatids, so as soon as they're separated, they go off in different directions. And again, what's going to happen here is dynesin moves that sister chromatids this way or the other way, and as they're moving, the kinetochore microtubule is disintegrating behind them. So now that kinetochore microtubule starts to shorten, and the sister chromatids go off in opposite directions. And that's what's happening in anaphase. So anaphase, basically, each sister chromatid moves towards the poles, and they move towards that paracentrioles that's or that centrosome that's at the end of the cell. And of course, like I said before, the polar microtubules are allowing the cells to elongate at that point, which means that you have now enough room in between those two sister chromatids to start to basically allow that cell to then separate from one another. Okay, so then the last thing that's going to happen is telophase. And telophase basically is where the chromosome movement stops. The chromosomes will start to decondense. The nuclear membrane will begin to come back to reform. The nucleolus is going to come back again. And then what happens in the middle of this cell basically is that you're going to form a cleavage furrow, which I'll show you in the next slide, or a metaphase ring. That's going to be formed by the actin cytoskeleton, and that will basically cause those cells to pinch off from one another, forming two daughter cells. Okay, so this is the cleavage furrow up here. That's formed by with a combination of actin and a little bit of microtubules, but mostly actin. What happens in plant cells is that you can start to then form a new cell wall in between the two new cells, and you can then separate off your two cells. Cytokinesis is the process of separating the cytoplasm at that point. It's a little bit of a separate procedure from telophase, but it happens at the same time. And then you move into the next G1 phase of the cell cycle, having two identical daughter cells at this point that are basically the same as the mother cell. So then you reform the nucleus, the chromatin decondenses, the nucleolus comes back, and you have two new cells. Okay, so that's our process. So that's the end of what I need to talk to you about now. I'm going to put this online for you guys to view, and then when you come to class, having viewed this, I'll answer any questions that you have, and then we'll move on and talk about meiosis. So thank you very much. Bye-bye.